Okay, now in this section we will we will try to understand cloning and how researchers get the DNA they want to clone. Now, gene libraries are made up of pieces of entire genome that are stored in plasmids or phages. Phages are those bacteria that infect the viruses that infect the bacteria are called as the phages, and the plasmids are the extra chromosomal DNA. Now, the problem is usually in eukaryotic DNA. The prokaryotic DNA does not have as many introns, but the eukaryotic DNA has a lot of introns that are the nonsense regions. So in the process of mRNA processing, when the DNA is copied by RNA polymerase to make RNA, segments of the introns are also copied. Later on, certain enzymes are used in which they will cleave segments of the introns and put all the exons together. So you have an mRNA that is shorter, and it also has the exact code of the DNA. Now, if this messenger RNA is extracted from the cytoplasm, which is pretty much done in many laboratory settings, then through synthetic process, the messenger RNA can be used to synthesize a synthetic DNA or the copy or the complementary DNA. And this complementary DNA would have the exact segments that you can see over here, the purple pieces. So there are lots of copies of this cDNA that are present all over the world, and scientists all over the world, once they synthesize or they get a segments of the DNA and they have figured out there's a gene for that code, they actually are put into a database. So eukaryotic DNA has many introns, so it makes many cDNA copies, and transcriptase is the enzyme that will do that. That will help to make those segments. So what do they do with the cloned DNA? The cloned DNA has many uses. For example, in the first picture up here, you can see that it is used for protein production. For example, in proteins, for example, insulin and human growth hormones or vaccines. You can clone different copies of the foreign DNA and manufacture the proteins that can be later used in therapeutic conditions. Another example that is shown to you in the middle is that of the altered organisms with useful traits, for example, pest protection or herbicide resistance or added nutritional value. And these are all examples of genetically engineered transgenic plants that can be used. And third one over here is a cloning host provides abundant DNA for study. So a source of DNA that can be used for gene regulation, gene function, and nucleotide sequencing. So determining the sequence of DNA is a process which can have a tremendous amount of implication once we understand the gene. What does that gene do? You can use the gene to study whether it's organically grown food or whether it's genetically modified organism, or whether it's a disease that needs to be studied, whether it's in plant or whether it's in animals. So many different useful studies can be done in the future. We will probably use biotechnology for many diseases such as cancers of all sorts, AIDS, and in which genes therapy will be mainly used to modify these diseases and to cure them. So here are some more techniques or more terms that are shown to you. Random shotgun sequencing. And the random shotgun technique is a technique that is used in which small pieces of gene of the genome are actually sequenced, and these sequences are assembled using a computer, and any gaps between the pieces have to be found and sequenced. So the sequence of, for example, entire viral genomes are now relatively easy to obtain. The genomes of yeast, E. coli, and over 70 other microbes have been mapped, and about another 100 are in process. So the Human Genome Project was a massive undertaking resulting in sequencing of the human genome. So that's the random shotgun technique. Random sequencing is also another process in which you are sequencing fragments of DNA from different. This is again a bacterial map that has been shown, and segments of the DNA have been randomly shared and amplified and pretty much sequenced. So let's go to the next slide. Bioreactors, they use bacteria to produce gene products, and some of the examples are shown to you on the slide. Insulin, that is used to cure diabetes or human growth hormones, and cellulase that break down the cell wall for animal feed, or factor VIII for hemophilia have been synthesized in here. Genetically modified organisms can be used especially for transgenic plants, and bioreactors are used for that. And again, it depends on many of the reasons. You know, you want to make a potato that is pest resistant, or you want to make soya beans that are resistant to common herbicide. Many corn and cotton and are modified for herbicide and pest resistance. So you are, in the U.S., we actually have a lot of genetically modified plants. And we also have, for example, the golden rice, which was one of the precursors of genetically modified plants. There were regions in the world, third world countries, they're deficient in production of rice and vitamin D, and we inserted those foreign genes into the rice crop. And here is a shorthand image of how the process is done. For example, here is the bacteria. The plasmid is extracted. The plasmid is cleaved by the restriction enzyme. You add the gene of interest. You put it back into the plasmid is resealed and placed back into the bacterial colony and amplified within that. And then you are inserting the transgenic gene carrying the foreign gene, and then that is grown to a new plant. Plasmid are just one vectors that can do it. We also studied the polymerase chain reaction that can also amplify the genes. Here is an example of gene therapy in which human gene materials are inserted, and we use a retrovirus. That retrovirus are viruses that insert into a gene and form. They're actually DNA viruses, and that can be used for cloning purposes. For example, up here on the first image, you can see the normal gene is isolated from a healthy subject. Gene B that is cloned, gene is inserted into a retrovirus. Bone marrow sample is taken from a patient with a genetic defect. So you've got the bone marrow sample. Bone marrows are infected with the retrovirus. The trans-affected cells are reinfused into the patient, and the patient is observed for normal expression because the retrovirus actually transforms the infected marrow cell, and this is a gene therapy that is used to do that. It's not very successful yet in many cases, but it's a process that is fairly recent, still about 5-10 years old, and has many promising outcomes for the future. So in this particular chapter, we will end up here. We learned a lot of different terminology and techniques. And why is it related to the microbes? Is because microbes were actually the first vectors that were used, which is the plasmid that was used, a vector from the bacterium, and they were more commonly and commercially acquired. So the vector, which is a plasmid, actually gave suggestions, or it gave information as to how different techniques would be revised in the future. And there are currently many proteins that are used for the recombinant DNA technology. For example, many of the immune treatments for interferons and interleukins and macrophage colonies are used to stimulate bone marrow activity after bone marrow grafts. Certain hormones such as erythropoietin, a peptide that stimulates bone marrow to treat some forms of anemia, factor A that is needed for blood clotting, to treat the hemophilic patients, relaxin and aid to childbirth, human growth hormone that stimulates growth in children with dwarfism or prevents wasting syndrome, enzymes such as antitrypsin, vaccines, for example, in hepatitis V, and to treat type B meningitis. Many experiments are done on malaria and AIDS vaccines recently too. And besides that, we are also studying them in bovine growth hormones and spider silk, which is a light, tough fabric for use for parachutes and bulletproof vest. So a lot of many different approaches to study genetically modified products using the recombinant DNA technology.